right, thank you so much and good morning. It's really my pleasure to be a moderator here for this exciting basic science session, the inaugural within the CCF annual meeting. And at this time, I'd really like you all to be introduced to Dr. Joel Schneider, who received his PhD in chemistry at Texas A&M University prior to moving to University of Pennsylvania as a postdoctoral fellow. After this point, he joined the faculty at the University of Delaware, and as a professor of chemistry and biochemistry at UD, he was recruited in 2010 to the National Cancer Institute at the Center for Cancer Research to serve as chief and to build their new chemical biology laboratory. In his independent research, he develops biomaterials for use in the local delivery of therapeutics towards the treatment of cancer. And his particular interest is in peptide-based hydrogels that are formed by self-assembly, and his work has established how material composition and structure influences material function, which lays the foundation to ultimately translate these materials into the clinic. Joel also serves as the Deputy Director of the Center for Cancer Research, as well as the past president of the American Peptide Society. So please welcome Dr. Schneider.
Thank you for the kind introduction. Let me start this morning by thanking the organizers and the chairs for their kind invitation to tell you a little bit about my science this morning. So first and foremost, my talk is going to be quite a bit different than most of you here. And I'm a chemist and a materials chemist, and in my work, we develop materials that facilitate the local delivery of therapies. And I'm looking for the clicker, if anybody has it. Here? No? There we go. Okay, thank you. All right, so currently in the lab, we're interested in developing three types of materials that enable the local delivery of therapy directly to tissue once implanted. And the first type of material is a material I'll talk to you about today in detail, and these are peptide-based hydrogels, which are formed from peptide self-assembly. The second type of material are target-oriented materials, and then the third are membrane-perturbing peptides, which we use to ferry drugs directly into cells. Now, the hypothesis that guides the research in the lab is we strive to understand the mechanisms by which all of these materials are formed at the molecular level. We also strive to understand how they're degraded once implanted in vivo. And importantly, we understand the underlying material structure to molecular detail. And this understanding then allows one to develop materials towards targeted clinical applications much more effectively. So today I'm going to tell you a story about work towards treating a type of cancer with our peptide-based hydrogels. Now, for this audience, I thought I might step back a little bit and describe peptide self-assembly to introduce you to the idea. So, peptides in general are small fragments of proteins, typically no more than 30 or 40 residues in length. They can be naturally derived, or they can be de novo designed. And some of these peptides have a propensity to assemble or to aggregate, right? And they can do so in two manners, right? You could have nonspecific association or aggregation that leads to amorphous aggregates, or you could have really well-defined self-assembly events that lead to very well-defined and distinct morphologies. And here I show a fiber that's formed from the self-assembly of A-beta, a peptide that's associated with Alzheimer's disease. Okay, now, whether or not a particular peptide sequence assembles in aggregates, it really depends on its exact sequence, its primary amino acid sequence, and also the environmental conditions that you expose that peptide to, okay? And we all know in this room here that peptide aggregation can be detrimental. It'd be associated with disease, for example, Alzheimer's with the self-assembly of tau and A-beta peptide. It can also be a big problem in the formulation of peptide-based therapeutics, right? But what I'll show you today is that peptide self-assembly can lead to materials that have functional utility, right? So, you know, now it's really well appreciated that peptides can assemble to give you different materials and different morphologies, right? But this had not always been the case. The first example of peptide self-assembly leading to a material was given in 1993 by Zhugang Zheng and Alexander Rich at MIT, and they published a paper where they showed that the 16 residue fragment isolated from the yeast protein zootin could self-assemble to form these fibrous membranes. And this is not a particularly nice-looking micrograph, but it is the first example. And after this paper was published, the field of peptide-based materials for drug delivery exploded. And today we know that you can either derive peptides from natural sources, or you can de novo design them to self-assemble into a myriad of shapes, right, including discs, spheres, tubes, tapes, ribbons, and even plates, right? And flowers, and even teeth, right? But the most ubiquitous morphology is that of the fiber, okay? And this is a privileged morphology, and that fibers can undergo higher order self-assembly to form networks of fibers that constitutes the formation of a three-dimensional viscoelastic hydrogel that encapsulates a large volume of water, okay? Now, my group for many years have been developing peptide-based gels for drug delivery, and we design these peptides de novo in such a way that when you initially dissolve them in water, they're monomeric and intrinsically disordered. However, you can design that primary sequence in such a way to be responsive to an environmental cue which triggers self-assembly. And when you trigger assembly, the peptide self-assembles into a network of fibers which constitutes the formation of a three-dimensional gel, right? And you can do this directly in a syringe. Now, if you have a therapy, whether it be a small molecule, a protein, a nanoparticle, even cells, they get directly encapsulated into that fibrous network with 100% encapsulation efficiency. Now, the materials I'm going to tell you about today have a really cool mechanical feature, and that is they shear thin but self-heal. So if you push on them, they flow, but as soon as you stop pushing on the gel, it reheals or sets up as a solid material. And this allows you to deliver these materials that have encapsulated therapies by syringe, or as I'll show you today, by spray. Okay? Now, once implanted in vivo, the rate at which those therapeutics are delivered to the local tissue is dependent on the physical attributes of that fibril network. Okay? And to date, we've designed peptides that will self-assemble in response to ionic strength changes, pH changes, thermal energy, light, metal ion binding, cell culture media, and even the action of some enzymes. This technology lets us then deliver drug-laden gels either topically, subcutaneously, directly into organs, as shown here in this mouse liver. We can implant them peri or intratumorally. This is an example of a xenograft where we use ultrasound to implant the gel surrounding this tumor. We can resect the tumor, and you can see the clear gel here totally surrounds the tumor mass, and we use these to deliver highly toxic chemotherapeutics to the tumor while avoiding systemic toxicity. Now, as I said in the introduction, we're really interested in determining the underlying structure of these materials and the mechanisms by which they form. I want to take you through a few slides that show that. Although we've developed and designed over a couple hundred peptide sequences to form distinct gels for our mechanistic and structural studies, we use a peptide we call MAX1. It's 20 residues in length, and we've designed this peptide to fold into this beta hairpin conformation when it's self-assembled in the fiber. It contains two beta strands here and here, which are amphiphilic, meaning they alternate in their residue composition with hydrophobic and hydrophilic residues. These two beta strands are connected by a tetrapeptide sequence here with this valine, diproline, proline, and threonine, which we've designed to adopt a type 2' beta turn. And this is a common beta turn found in naturally occurring proteins. Now, we've gone ahead and solved the structure to molecular detail of the fibers that are formed by this peptide in collaboration with Rob Tico at the NIDDK at the NIH using solid-state NMR. And here I show a fiber, only 8 copies of the peptide. The long axis of the fiber would be in the plane of the board here, right? And you can see that although this peptide is unstructured before you assemble it, once it assembles, it adopts this targeted beta hairpin conformation. So if you follow this light blue strand, right, it's a beta hairpin. The peptide self-assembles to form a bilayer of cross beta sheet structure. So the front monolayer is in greens and blues, and the back monolayer is in pinks and reds. Now, if I pick up that fiber and I turn it into the plane of the board, and we look right down the long axis of the fiber, you can see these valine side chains and CPK representation here packed really nicely in the interior of the fiber. And that drives self-assembly of the peptide, the formation of those hydrophobic interactions. The exterior of the fiber contains the lysine residues, which are well hydrated. Now, the formation of this bilayer here represents the entropic driving force for self-assembly, and that it excludes all of the water from that interface. And that is the thermodynamic driving force for gelation, right? The release of ordered water from that unstructured monomer. All right. Importantly, the fibers that are formed from MAX1, this peptide, are monomorphic. That means they self-assemble to form one morphology, one type of fiber. Okay, and this is really distinct from all the disease-forming peptides that you've heard about, A-beta, right? They'll self-assemble in a myriad of ways to form different types of fibers. Because these self-assemble to form one distinct morphology, we can then use structure-based design protocol to design materials at the molecular level because we know where every atom is in the fiber and in the gel, okay? Now, the way these things self-assemble is that they're initially intrinsically disordered and monomeric. Once you trigger self-assembly, they rapidly collapse and assemble into a family of oligomers where each of the peptides is largely unstructured in these spherical objects, right? These grow with size over time until you reach a critical size that nucleates the folding and the self-assembly of the peptide to form a little cluster of fiber, right? These fibroclusters nucleate throughout the entire container in which you're forming the gel. Each cluster has a little dangling end of fiber that interpenetrates its neighboring cluster until you percolate the entire volume of the container. That, by definition, is the gel point. And as time evolves, so does the network until gelation is complete, all right? Now, with this structural and mechanistic understanding, we've gone on to design over 200 primary sequences that self-assemble to form distinct gels that allow you to deliver distinct therapeutics. And so we have gels that can deliver different types of small molecules, protein therapeutics, nucleic acid therapeutics, nanoparticles. We have cytocompatible gels that you can encapsulate, for example, stem cells in 3 dimensions, grow those cells in the gel matrix, and then simply shear thin deliver those cells back in vivo. We have designed gels that are inherently antibacterial that we've coated medical implants with. We understand enough about this system where we can tune the mechanical viscoelastic properties to molecular detail. This has allowed us to develop surgical tools, for example, gels that enable or facilitate microanastomosis, which is the suturing of ultrafine blood vessels and lymphatic vessels. But today's story I'm going to tell you is about the development of a gel that we're developing towards the treatment of mesothelioma, which is a complex surface cancer that primarily occurs in the pleural cavity, pleural lining that separates the lung from the chest wall, okay? So this isn't cholangiocarcinoma, which we're all here this week for, but I'm hopeful that this example might spur some ideas on how we can develop materials to deliver therapies that might impact this community here. Now, mesothelioma is a complex surface cancer, and this is in contrast to many solid tumors that grow spherical masses where resection is facilitated to leave relatively clean margins. When you resect mesothelioma, you typically always leave behind residual cancer, okay? And so recurrence is a big problem. Now, on the left, I show a cartoon of the cancer. In yellow is the sheet-like tumor growing in that pleural cavity that separates the lung and the chest wall. On the right is a picture taken by my collaborator, Chong Hong, who's a thoracic surgeon at the NCI at the Clinical Center. and you can see the tumor growing as a sheet of these nodules in the pleural cavity separating the lung and the chest wall. This is a particularly bad cancer that's infiltrated into the chest wall, into the lung. Now, in addition to removing these tumors, Chong also is interested in developing therapies and trying to understand the molecular basis of the cancer. And he did an analysis which showed that patients that express higher levels of a particular microRNA, microRNA 215, live longer than patients that express lower levels of that same microRNA. And moreover, he went on to show that microRNA 215 is responsible for targeting genes that facilitate the progression of the cancer, okay? So he and I got together and decided we would try to develop a material that could deliver microRNA 215 to patients as part of the resection therapy, okay? So an adjuvant to the resection therapy. And what I'm going to show you is that we designed a gel which we call surface fill hydrogel that does just that, right? So when this tumor is removed, the surgeon gains access to the tumor through the chest wall, a lung is deflated, and then the sheet-like tumor is removed. This gel can be sprayed onto the lung, into the chest wall cavity here. Once implanted, it's a shape-changing gel, so that when you reinstate the native anatomy of the lung and the lung blows up, it can move and push the gel out of the way and into the crevices and nooks and crannies of that freshly resected tissue, okay, to facilitate better delivery. And what we're delivering here are nanoparticles that are comprised of intrinsically disordered peptide, amphiphilic peptide, that's bound to that microRNA 215. And as I'll show you today, these particles are taken up by residual cancer cells by endocytosis. They escape the late endosome to deliver their microRNA payload to knock down and attenuate the genes that facilitate the progression of the cancer. Okay, now, we're going to get a little chemical here, so please forgive me. Or you can have a cup of coffee and take a nap, but it's up to you. So the first step in developing this material is to develop the nanoparticles, right? And I'm a pretty simple guy, and my first idea was to simply take a polycationic amphiphilic peptide, of which we've had many in the lab, and see if we could get it to bind to microRNA through electrostatic interactions, right, to make the nanoparticle. And we went to the refrigerator and we pulled out 3 peptides, and this is the first time I've ever been lucky in my scientific career. The first peptide we tried bound in a cooperative manner, which we were looking for, with a binding association constant that wasn't too tight and wasn't too loose. And that's this peptide 1 here, the red data here. Very nice cooperative binding to the microRNA. DLS and cryo-EM showed us that this binding event led to particles that were spherical in nature, that have a diameter of about 200 nanometers. And importantly, we could modulate the charge or the zeta potential of these nanoparticles by simply changing the mole fraction or the stoichiometry of the positively charged peptide we use to bind to the negatively charged microRNA. So we can make nanoparticles that are electropositive all the way to electronegative. And we tested each one of these particles' ability to enter cells, and I'll save you that data. This 10 by 1 particle here, which is electropositive, and the 10 by 1 here, this corresponds to the number of the amines on the binding peptide to the number of phosphate groups on the microRNA. But this electropositive particle gets into cells quite well, okay? How well can you deliver microRNA using these particles? Just as well as lipofectamine and better than Darmafet and in vivo jet polyethylenamine, and these are all commercially available vectors. Now, once delivered to the cells, they are taken up by the cells by clathrated mediated endocytosis, as these flow experiments have told us. And once in the cells, they can knock down the targeted genes quite well, okay? And this knockdown corresponds then to an inhibition of cell growth in this 2D colony forming assay using mesothelioma cells. And we've also got 3-dimensional cell growth data here that I'm not showing. Now, with the nanoparticles in hand, the next step was to get them encapsulated into the gel, right? So this is easy. We take one of our self-assembling peptides. We simply trigger self-assembly in the presence of the particle. We can encapsulate those particles with 100% encapsulation efficiency. And here's a cryo-EM micrograph that shows you these nanoparticles encapsulated within the network of those fibers that constitute the formation of a gel. We can release those nanoparticles at a slow, sustained release rate, slightly dependent on the weight percent of the peptide used in the gel formulation. And this is rheological data that just shows you that when you encapsulate a large number of these particles in the gel, it doesn't affect the rheological or mechanical properties of the gel to be shear thin delivered, okay? And this is important because this then allows us to spray these gels, which is shown here onto a, and this is just in vitro model, which is spraying onto a pig lung. We've dyed the gel purple so you can see it. We expand the lung, and what you can see is that the gel is shape changing. It accommodates large volume changes in the lung, right? Then it stays put where you introduce it. Okay, now we've shown efficacy in 4 models of mesothelioma. What I'm just going to end with today is to show you a recurrence model from a resection experiment that we did. So in this experiment, we take groups of mice, we implant mesothelioma primary cells, and we let them develop into palpable tumors. It takes about 5 weeks, after which we resect those tumors. We have one on the left flank and one on the right flank. And on the left side, we introduce sham gel, which is delivering nanoparticles that contain scrambled microRNA. And on the right flank, we're delivering therapeutic microRNA 215. Okay, and in addition to microRNA 215, we're going to also deliver microRNA 206, which is another therapeutic microRNA that my collaborator had discovered. And what you can see, let's take a look at the left column here. Here at day 0, you see both of the tumors. We resect, we inject the sham gel and the therapeutic gel on the left and right sides respectively. At day 4, everybody's happy, there's no recurrence. But at day 28, you see reoccurrence on the sham side. Very little reoccurrence on the therapeutic side. The same can be seen with delivering microRNA 206, right? This data here is quantitated here, which you can see one single injection of the surface fill hydrogel delivering these nanoparticles can greatly attenuate reoccurrence of the cancer. So I'm going to end with that and just tell you that currently in the lab, Chung and I are translating this to clinical trials at the medical center, and we're currently working out some issues with the FDA from a pre-IND meeting we had with them not too long ago. So with that, I'm going to end. I'm going to thank a small group of highly talented fellows at the NCI. My lab is funded by the intramural program at the NCI. I'd like to thank my collaborators, especially Chung, you for your attention, and I'll be happy to answer any questions at the end of the session.
Dr. Schneider, thank you so much for a wonderful talk. Our next speaker is Dr. Gregory Lesinski. We heard a little bit about Dr. Lesinski's background, just a little bit on his research focus. His program is interested in understanding the interactions between the host immune microenvironment and tumor cells, and using that information to develop novel therapeutics such as adoptive cell approaches to augment immune-based therapies. And the title of his talk today is Novel Adoptive Cell Therapy Approaches for Advanced Solid Cancers. Thank you, Dr. Lesinski.
All right, well, thank you for the introduction and for the opportunity to share the work that we have going on at Winship in Atlanta. I'll just jump right in, and I borrowed this very nice summary from Dr. Gretton to illustrate the fact that in the setting of cholangiocarcinoma, there's a lot going on in the space of immunotherapy. And we're all familiar with the application of immune checkpoint blockade along with various combination approaches other agonist checkpoint antibodies, vaccines, bispecifics. There's many things occurring in the active space, both in the preclinical as well as the clinical setting. And the focus of what I'm going to talk about today is in the setting of giving cells back to patients, in many cases animal models, as we have a lot to do. And really, to sort of zoom out and envision where we're at as a field in cholangiocarcinoma and other biliary tract cancers, this data from the TOPAZ-1 trial really established that immunotherapy is most likely to be given upfront to patients together with chemotherapy. And what I would argue is that given this shift in treatment paradigm, it's really going to change the population of patients that will come in to receive subsequent therapies. From an immunotherapy standpoint, then, I feel that we have to be a bit more aggressive. Many patients will have PD-1 resistant disease, so it's less likely that putting a PD-1 based combination up front after they've progressed on this therapy will produce game-changing results. And this is really changing the landscape of patients that will enroll in these trials, hence the need for adoptive cell therapy approaches if we can get them working. And one of the things that I've really enjoyed over the last 7 or so years since entering the cholangio space has been the idea that we can work across institutions to make an impact in a relatively rare tumor. And this slide illustrates some of the collaborations that we've been fortunate to be a part of, both within cooperative group settings, together with the foundation, of course, and then with other institutions to drive this effort forward. And this picture illustrates one of my fast run times at our institutional 5K with my son and his friend. And his friend's mom is actually a family friend of ours who's a near 15-year cholangiocarcinoma survivor treated at Emory. So we have our own local inspiration here in addition to all of you. So really a pleasure to be a part of this group. Within Winship, we have a very active GI oncology translational research program that comprises researchers in the basic science setting as well as the clinical setting. And our overall goal is to integrate the findings from the laboratory and translate them into patients. One of the things that we're most proud of in this space is that we have really lured many individuals, perhaps some of you here today, who normally didn't work in the setting of cholangiocarcinoma, and then gotten them interested in GI cancers of the bile duct, pancreas, and other sites. So this is something that I think you need to infuse new ideas to make advances, and we're trying to do that here. We're trying to do it at our institution, and it really can be a recipe for success. So what do we want? We want to impact cholangiocarcinoma with adoptive cell therapy and have impacts in the setting of more survivors. People like Melinda, who's been remarkably involved in the organization, is a prime example of a success story, and where giving CD4 specific T cells back to a patient can impact their clinical outcomes. And this was one of the earliest publications from Dr. Tran while he was still part of Steve Rosenberg's group. Now he has his own research operation, but indicating that you can take a CD4 T cell and you can give it back to a patient and you can elicit dramatic clinical responses. This really laid the foundation philosophically for much more work that needs to be done. And before we can really dive in and go forward, it's always helpful for those that don't work in the cell therapy space to rewind a little bit and think about where it all began. And I'll lean on my friend, Dr. Christian Capitini from the University of Wisconsin, who gave us a bit of a history lesson at the recent winter school for SITC, and I'll share a very brief highlight of that just so that we can all be oriented. So adoptive cell therapy, where did it all begin? Really several decades ago. This idea was born out of observations from donor lymphocyte infusions, where bone marrow donors were given cells to treat patients with other conditions, viral infections, and relapsed leukemia. And one of the key observations from this early experience was the fact that depending on the number or the percentage of T cells in that donor product, there was a concurrent increase in the observation of graft versus host disease. So there was this immune reactivity that was going on, and at times were very dramatic clinical remissions observed, especially in the setting of CML, where myeloid leukemias were quite responsive to this on occasion. The field took another turn and another advance in the 70s when the cytokine interleukin-2, formerly known as T-cell growth factor, was identified as a substance that you could put on and selectively expand and grow and empower these T cells for therapeutic intent. And Mike Lotz, who was at the NCI, published these two papers with Dr. Rosenberg's group in 1980, where they showed you could take out peripheral blood cells, put them with IL-2 as an exogenous factor, and differentiate them into what were known as lymphokine-activated killer cells, or LAC. And these had potent anti-tumor activity in vivo. And then Dr. Rosenberg later translated that idea of LAC cells derived from the peripheral blood, bathed in IL-2, then put back into patients, and showed dramatic clinical responses with this individual, a melanoma patient in her 30s, who had a complete response after re-administration of these cells intravenously. So very potent stuff here that we're talking about. The field later advanced when the idea of LACs transcended into taking actual tumor cells out and then selectively growing and expanding out, with the help of IL-2, T lymphocytes that would react against the cancer. So this idea of TILs, or tumor-infiltrating lymphocyte therapy, was really born out of this work and many advances before it as one of the earliest examples of a successful adoptive cell therapy. Other advances in this space indicated that, speaking of space, if you make space in the peripheral blood by lymphodepleting a patient or an animal patient, you could actually get the T cells to expand more effectively and then create these super armies marching around in the body to go in and selectively eliminate the cancer. And this is not little stuff. It can be quite toxic, but cyclophosphamide is typically given in patients. In animal models, people will fluctuate between this chemical means as well as total body irradiation in order to make that space. But indeed, it seems to make the T cells more effective at their expansion and then efficacy and persistence upon in vivo transfer. So where are we at today, or really just a few years ago from this review? The field has really expanded. It's exploded, in fact. And all the different T-cell therapy and other cell therapy modalities are listed vertically along this axis. You can see the rapid expansion from the years 2020 on to 2022, and this continues to go on today. And there's a variety of different therapeutics ranging from antigen-presenting cells to cytokine-induced killer cells, TILs, CAR T cells, many others that are at various stages ranging from preclinical development all the way into patient phase 2 and 3 clinical trials. So this is definitely the future and isn't going away. And the task for us as a field is how do we best apply that to this setting in cholangiocarcinoma. So a little more detail on one very widespread example of adoptive cell therapy is this idea of chimeric antigen receptors, or CAR T cells. The way this process works, for those that don't live in this space every day, is you can take a T cell and you can artificially engineer it to recognize any antigen you want with a little bit of viral technology that we'll talk about. So typically, patients are leukapheresed. You get the blood product from the periphery of the patient. You can then engineer them in the lab using a viral transduction that engineers the cell's specificity for a tumor antigen. You can expand and grow these out in the millions in the Petri dish under GMP conditions, and then you can infuse that product of redirected T cells into the patient, where hopefully it will act as a smart bomb to go after and cytotoxically eliminate those tumors that express the antigen that you want. So this is the overall scope of this approach. This is what a CAR, or a chimeric antigen receptor, looks like. It's sort of this hybrid molecule that has different variable heavy and light immunoglobulin components forming a single chain FV on the surface that will selectively recognize components of antigens that you want the cell to recognize. There's a spacer region and then different signaling flavors that are downstream through the membrane that really facilitate T cell activation and survival and make the whole thing go. And an important thing to note is that this virus that's engineered into the T cell, it is essentially removed from its replicative potential but it does infect the T cells and offer this new antigen specificity to it. So this is really the secret recipe for endowing them with tumor-directed targeting. So what are the limitations? Why don't these things work, particularly for solid tumors like cholangiocarcinoma? A number of things can happen that will thwart their activity. The first is the tumor losing the antigen that you engineer a CAR or a TCR transgenic T cell against. This is a known mechanism of resistance. The second that many are trying to overcome is this idea that once you put the T cells in, they go away. They'll either become exhausted and less functional, they can enter sites where the tumor microenvironment is dominant, they can then be forced to undergo apoptosis. Many of them can't even get there. They'll have impaired trafficking to the tumor because of these disruptive chemokine gradients. Finally, there are appreciated redundant mechanisms of immune suppression that exist within these aggressive tumors, like myeloid suppressor cells, T regulatory cells, immune stimulatory cytokines that don't necessarily favor cellular survival if you're a CAR T cell. So the other thing to not gloss over is the toxicity risk. There can be certainly low or abundant at times expression of the antigens that the cells may be directed against on normal tissue that could lead to toxicity. There could be cytokine release syndrome that manifests in a severe inflammatory systemic response seen across heme malignancies. So all these things are ones that you have to consider when you're doing the early phase trials with adoptive cell therapy. So as a group, our intent is to figure out what we can do to offer solutions to these problems. And the first solution that we're going after, and we'll talk a bit about this, is the idea that if you change the starting template of the T cell, you can do better. Also, we can diversify our antigen targeting, engineer the T cells in a variety of ways to resist exhaustion, and then, of course, pursue combination therapies. This is a review from Hannah Knockelman, who's a former MD PhD student, now a resident at Stanford in the Palos laboratory at Emory. She wrote this wonderful review about all the different ways you can use T cells as a new engineering template and overcome many of these limitations. Going from left to right at the top, you can see that you can do new generations of intracellular signaling domains to offer the T cells advantages. You can engineer them to traffic differently, and you can even engineer them to express other cargoes like cytokines or anti-tumor antibodies that can be concurrently secreted as an armored CAR, or then even more sophisticated, employ Boolean gating and other logic models to the T cell itself so that it can recognize only the antigen you're targeted, but then if it sees a self-antigen, it will shut it off, affording a different safety switch. So there are a lot of different ways that you can play with the T cell and make it do what you want to do, and those studies are ongoing. So our group has been fortunate to apply CAR T cell therapy in the setting of solid tumors like pancreatic cancer, and this is a CAR T cell approach targeting mesothelin that could, of course, be adapted to mesothelin-positive cholangiocarcinoma as well. And this is a multi-group collaboration between our lab that brings the GI cancer expertise, that of Dr. Crystal Palos, who's a CD4 T cell expert, and then Dr. Ned Waller, who is a bone marrow transplant doctor with interest in GI cancers and heme malignancies. Our team also has a medical oncologist, Dr. Tunji Alashe, our statistician, Dr. Yuan Liu, and then Dr. Carl June at UPenn, who has been very collaborative in providing us with vectors and his institutional experience in adapting these cells. So what problem does this program overcome? This is really focused on the first solution, and that is changing the T cell template that we use. So Crystal's lab has discovered a novel T cell population expressing a surface marker called CD26 that marks these cells that have a potent polyfunctional cytokine producing ability that when transferred to a tumor-bearing host can selectively identify and eliminate tumors. And some earlier published work from individuals in her lab, Stephanie Bailey and Michelle Nelson, illustrates the potent ability of these cells. If you look on the left at these flow cytometry plots, you can appreciate that these cells can currently make both IL-17, interferon gamma, as well as other cytokines like IL-22 that really identify them with a unique phenotypic property to provide help and even at times potentially eliminate the tumor cells themselves when they're redirected with antigen. So when you engineer the cells with targeting mesothelin, you can see on the right side that animals bearing tumors are eliminated upon adoptive transfer of these cells, and that tumor control is superior to doing the same thing with T cells that have been polarized to other Th subsets like Th1, Th2, or Th17 alone. So a very, what we consider an ideal cell template for subsequent engineering. We've then taken another approach at, while the cells are expanding, treating them with various pharmacologic inhibitors to make them more ideal. And what we're calling the secret sauce was these observations born out of treating the T cells during expansion with a PI3 kinase inhibitor, where you can see their mitochondrial membrane potential increases, the actual expression of the CD26 molecule on the surface increases, and then importantly, they have more stem memory properties expressing TCF1 and less TIM3. When we've done pilot studies and infused them into mice bearing orthotopic pancreatic tumors, we can see that the cells persist much better and reach the tumor compared to the controls. And this work was done by Guillermo Rangel Rivera from Dr. Paulos' lab and Ronnie Funk from the Waller lab. So our overall model for these CD26 cells is that we think they'll solve two particular problems. One, their polyfunctional cytokine producing properties and their cytotoxic capacity can theoretically get into tumors and persist in the host more effectively. Second, the CD26 molecule, in addition to being a marker on the surface, itself has functional properties. Specifically, it can enzymatically truncate various chemokines at their N-terminus that are involved in the myeloid dominant phenotype of pancreas and many of these other GI tumors. So some of the enzymatic substrates for CD26 are things like GM-CSF, SDF1, many of the factors that are really pro-myeloid cell, again, a dominant force in these desmoplastic stromal tumors that we need to compete with if we're putting an adoptively transferred T cell in there. And then ultimately, we have plans, and now we're fortunate to have funding as well as institutional support to do a first-in-human trial, infusing either these cells alone or co-infusion of these cells together with a conventional meso-targeted CAR T cell that, through collaboration with Carl, are differentially barcoded so that we can track their fate in vivo. So other approaches of adoptive cell therapy. We've talked about CARs. There are, of course, TILs, TCR engineered T cells, engineering T cells to have more central memory properties or recognizing viral antigens. Many of these are having merit in wide open places. So just a bit, the difference between a TCR and a CAR, the T cell receptor has the natural endogenous signaling machinery. So although it has the limitation of having to recognize an antigen in the context of MHC, it can have a more natural downstream signaling molecule as compared to the CAR that really just has the antibody component to it. So TILs were recently FDA approved out of melanoma, where you can see compared to ipilimumab, an immune checkpoint inhibitor, the TIL therapy in patients with advanced melanoma had superior overall survival outcomes as well as progression-free survival in the setting. So Iovance has now marketed that product. And then where are we going as a lab? I'd like to point you out to some of the work in depth and visit the posters, see the talks from our group at Emory. Dr. Emily Warren is a proud recipient of a fellowship from the Cholangio Foundation, where she can implant orthotopic cholangiotumors into mice, in some cases image them, and then she has found that we can identify CD4 cells that express CD26, our molecule that marks these good T cells, and then our goal is then to develop novel animal models where we can then put them back and hopefully elicit efficacy leading to trials and, of course, get more survivors. Megan Whitling from the Paulos lab is also setting ourselves up for success in better understanding the immune landscape of these tumors. So in similar models, she has characterized what these tumors look like, what the T cells are in other cells, and then indicated that CD4s persist over time as CD8s go away. And what we're looking at then is this natural predilection towards CD4 T cell dominance in the tumors, and can we take that and make more effective cell therapy products for cholangio? Then finally, Dr. Francie Bennett, a surgery resident within our group, collaborating with the Palos Lab, and Dr. Mythel, of course, she has taken to actually generating those TILs and demonstrating that with different polarizing cocktails, we can expand them in vitro and then have cytotoxic activity against cholangiocarcinomas in enclosed systems. So please check out all their posters, see their talks, and you'll get a better flavor of where we're going with our program. Just to conclude, we're applying adoptive cell therapy approaches ranging from CAR Ts to TCR and TIL approaches, and certainly there's other cell therapy products out there. And our goal is then to identify more robust and relevant preclinical models that can get us data to accelerate clinical testing of these platforms. So my acknowledgments here, especially to patients and their families for enrolling in clinical trials, my lab, the Palos Lab, Dr. Mythel, and many collaborators across the continent that have helped us and do this work. So thank you for your time and attention. Sorry I went a minute over.
Our next speaker is Dr. Dieter Sauer. He's a physician, scientist, and professor at the German Cancer Consortium. He heads the Division of Translational Cancer Research at the German Cancer Research Center and is the director of the Institute of Experimental Cancer Therapy. His group studies fundamental biologically and clinically relevant aspects of gastrointestinal cancer. How does it develop? How does it progress, spread, and why it becomes resistant to anti-cancer therapies. Over the last 20 years, his lab has generated unique biological resources and tools to analyze the therapeutic response of cancer subtypes in vitro and in predictive immunocompetent in vivo models. Thereby, the lab explores new ways to target solid tumors more efficiently. Dr. Sauer is quite prolific and has published over 190 papers and has received awards, including the German Cancer Award for his work. And the title of his talk today is Perihilar and Distal Cholangiocarcinoma, the Neglected Model. Thank you so much for coming all the way from Germany.
Yeah, many thanks for this very nice introduction. Thanks a lot for the invitation to this great conference. I find it really unique, this very special form of patient involvement, which I think is very important. I never saw that before. So very nice experience for me. Yeah, we will switch a bit more into preclinical models, and that's what we are working since about 20 years. And we came into the cholangiocarcinoma field a bit by kind of an accident, because we wanted to analyze tissue specificity of oncogenes. And as you know, I'm just now the pointer is, there's no pointer, no. But as you all know much better than me, we have different kinds of cholangiocarcinoma, so we have different anatomy, and we have, oh thanks, that's very kind. How does it work? Sorry. How does it work? Thanks a lot. Okay, so now again, so we have different anatomical sites. So we have the intrahepatic cholangiocarcinoma, but we also the extrahepatic one, the perihilar one, and then the carcinomas that arise in the common bile duct. And as you see here, they differ in their occurrence. So 10 to 20% are intrahepatic. About the vast majority is these perihilar ones, and the distal ones are at about 20%. And in contrast to these numbers here, we have very, very few extrahepatic cholangiocarcinoma data in our studies in the databases. So this is from CBio portal, and you can see that only about 5% of all sequenced tumors are in the end extrahepatic ones. So there's a bit of a lack of, on the one hand, genetic data and molecular data about these tumors, but also in terms of models, because almost all models that have been published so far are intrahepatic cholangiocarcinoma models. And then we came up with an idea that we know that there are tumor suppressors and oncogenes that are mutated, and these give rise to genetic syndromes, and these persons have an increased risk of cancer development, but this is always very tissue specific. So if you have an APC mutation, you will get most likely a colorectal cancer, but not other cancer types. And we were interested in the basis of that. So what makes a tissue susceptible to cancer development? And what we knew already, of course, that oncogenic signaling is very, very context dependent and also tissue dependent. And therefore, we decided to activate different oncogenes in different tissue types. And as you can see here, that can be done by the Cre-Lox-P system. So we can use albumin Cre to activate something in the liver. It's shown here by a fluorescent reporter. So this liver cells are in green. Here in the pancreas, so the cells turn blue if you express a laxet. Again, the green fluorescent protein here for the large intestine and the small intestine. But with one of our models, the PDX-Cre model, we got suddenly expression of Cre in several different tissue types. So we observed it in the pancreas, which was known very well, but we also got expression in the gallbladder and in the common bile duct, but not in the liver in the end, and in the duodenum. So we have in the end 4 different tissue types where we can activate an oncogene. And we validated that also then by another reporter line, and we looked at the recombination and the activation of this fluorescent protein, and we could see that this is really the biliary epithelium and the peribiliary glands. The basis of that is that we really can target the extrahepatic biliary system and not the intrahepatic one. This is based on development, because the extrahepatic biliary tree develops from the ventral pancreatic bud, whereas the intrahepatic biliary tree develops from the liver bud. So the cell of origin is different. And PDX1 is one of the transcription factors which is active in the progenitor cells of the pancreatic lineage of the ventral pancreatic bud. And as you can see then here, therefore all the cells and the organs that develop from this bud in the end are recombined by our PDX1 Cre line. So in the end, and then later on these cell types fuse or the organs fuse in the end. And the main message is though that the intrahepatic biliary cells derive from the liver progenitor cells, whereas the extrahepatic ones from these PDX1 positive cells. And then we activated a variety of different oncogenes in all these different organs. And what we observed, which was a bit expected, in the pancreas with PTF1A, we get pannin lesions and full-blown pancreatic cancer with KRAS that has been published before. And we could show that this is also the case if you activate oncogenic PI3K signaling in this organ. And the survival is quite similar of these animals then. However, when we did it now with the PDX1 Cre line, so also in the bile duct, then we observed a completely different picture. So we saw that in the PI3K model, we specifically got bile duct cancer, extrahepatic bile duct cancer, and that's shown here, in addition to pancreatic cancer, as you can see here. So these are pancreatic cancer and bile duct cancer in the same animal, and here just bile duct cancer, extrahepatic bile duct cancer. Whereas in the KRAS model, we just got pancreatic tumors as well as some animals do not develop tumors here. Then we looked also at gene dosage. So if we increase the gene dosage by increasing by breeding this oncogenic allele homozygosly, we can accelerate tumor development. We get very undifferentiated but also very differentiated tumors. So there seems to be a strong tumor evolution impact on these tumors, as well as the precursor lesions, the classical precursor lesions of extrahepatic bile duct cancer, these bilin lesions, as you can see here in the PRCK model, but in the KRAS model, we got just papillary hyperplasia. We wanted then know, is this really PRCK signaling and classical PRCK signaling? So we and PRCK signals via PTK1 to AKT, and then quite a variety of other pathways. And therefore, we deleted PTK1, and thereby we could really block tumor formation in the bile duct completely, and block also bilin formation in these animals. So, of course, then we were interested in what is the mechanism, or what is the basis of this different susceptibility of different organs towards different oncogenes. And it's nowadays a bit the common topic. If you don't know what the mechanism is, a genetic screen is always good, because thereby you can identify the genes that drive the process. And what we did is we performed an in vivo genetic screen using a transposon model. Transposons are mobile elements that jump through the genome from chromosome to chromosome in the end, and they can activate genes via a promoter here. They can also inactivate genes because they have a splice acceptor and a polyadenylation cassette. And depending on the direction how they jump into the genome, they inactivate or activate genes. But they can also do epigenetically rewire the genome by disrupting enhancer regions, for example. We used now this model in our PRCK bile duct cancer model, and could show that this really accelerates tumor formation in these animals. And then by sequencing the tumors and the integration sites of these transposons, we were able to identify the genes that are activated and inactivated. And you can see this is quite a lot, with a high number of alterations. And when we put all these genes together into a network, so this is a string pathway analysis network, then we see that many of these hits converge to P27, this is CDKN1B, as well, of course, PRCK, and also P10, which is the major regulator of PRCK signaling. And subsequently, we of course wanted to validate, is P27 really a major tumor suppressor in the extrahepatic bile duct, and therefore we deleted P27 in the PRCK model, and we could accelerate tumor formation quite substantially. But also in the KRAS model, where we do not get these full-blown tumors, we could suddenly induce tumor formation in a gene dose-dependent manner. So the heterozygous animals develop already cancers, and the homozygous then very quickly. And you can see that now we get a shift from pancreatic cancer to extrahepatic bile duct cancer, as you can see here. So, and this is, and then of course we looked into the human situation. So do we see that? Because many tumors in the human setting have a KRAS mutation. Very few have a PRCK mutation. Some have a P10 mutation. So the PRCK pathway is activated often by other means. But of course there are also KRAS mutations. And what we then observed was that P27 is downregulated in almost all of these tumors. It's not by genetic inactivation, it's most likely transcriptional or post-transcriptionally regulated, as you can see here. And also, and in addition, the downregulated tumors have a worse prognosis versus the ones that have high P27 expression levels. What is now the mechanisms of P27 regulation by PRCK? We see that very clearly in the tumor, so P27 goes down. Only in the PRCK model, with the KRAS mutation, you have still high levels. If we block in cellular systems PRCK signaling, we can upregulate P27 again at the RNA level and also at the protein level, as you can see here. So P27 is really a directly downstream target of PRCK signaling, and the mechanisms how it accelerates tumor formation is in the end that it bypasses oncogene-induced senescence in the bile duct, because we see that in a P27 wild type setting we get oncogene-induced senescence, which blocks tumor formation, whereas in the P27 knockouts we can overcome that completely. And to summarize these data, these genetic screening data, we have often alterations in the PRCK pathway. We see as genetic hits regulators of the PRCK pathway, P10 for example, that then activate AKT. We see dysregulation of P27, and finally, and all this converges in the end in dysregulation of the cell cycle and activation of proliferation and bypass of oncogene-induced senescence. This is now the model that we think, of course, both oncogenes can induce tumors, but we need secondary genetic alterations to induce these tumors. We know that for KRAS, we need a downregulation to switch them from the pancreas specificity of tumor induction to the extrahepatic bile duct, whereas for the PRCK driven model, PRCK itself is able to downregulate P27, and therefore we get this huge induction of these tumors. Apart from what I've shown you, which is mainly already published, I wanted to show you a bit of an outlook what we are now doing, because we became very interested in this tumor type and think it's especially with our new model, we can now much better also develop novel therapies in bile duct cancer. So what we did in the past years, we collected quite a lot of different patient-derived models from intrahepatic tumors, but also from extrahepatic ones, and I think especially the extrahepatic ones are really important because we do not have a lot of models. We have now 4. It's also not a lot, but it's a starting point. I think it's quite interesting that these tumors have a high mutational burden, the extrahepatic ones, which is not always the case for the intrahepatic ones. And we see also PRCK alterations in these tumors. And we want to use these organoids now for drug screens, as well as specific genetic models which we generated with our mouse models. For example, an INF43 mutant one, because we observed a lot of integrations in our transposon screens in the INF43 gene, and we could show, as you can see here, that INF43 is an important tumor suppressor in this tumor entity. And now we generated organoids from these models as well. We see that proliferation is increased in the INF43 knockout tumors. And what we then do is that we do drug screens in these organoid systems. So we have a collection of 411 drugs that target quite a lot of different pathways. And what we observed is if we lose INF43, then these tumors become much more resistant to therapy, as you can see here. But they have a very distinct vulnerability. So cell cycle drugs that target the cell cycle, for example, or epigenetic drugs are much more efficient in the INF43 deficient tumors, whereas quite some metabolic drugs or or drugs targeting protein kinases are more efficient in the wild-type tumors. We also worked on our animal models in the past, so we generated models where we can inactivate the driving oncogene by the Krelog-P system for KRAS, where we can excise the exon 2, the mutated one, or excise the PRCK mutated form. And what we then did with these models, with the PRCK model, we are waiting now until the tumors develop, and then by an inducible genetic system, we can inactivate the oncogene, and then look what's going on with these tumors. Do they get resistance? How does the microenvironment change it? And these are some in situ sequencing data where you can nicely see that these tumors respond quite heterogeneously to such an inactivation of the oncogene. In some of the areas, we get a lot of infiltration of CD4 and CD8 T cells. But in other regions, as you can see here, which are very well differentiated, as you can see by these epithelial markers, we do not see that at all. And this immune cell infiltration is accompanied here in this setting interestingly by TGF-beta activation and expression, which we find quite interesting because TGF-beta is considered as an immunosuppressive molecule, of course. So we think by that we can better understand what's going on in the microenvironment upon therapeutic intervention or genetic perturbation of the tumors. But in order to look at different susceptibility or response to therapy and development of resistance, we think we also need to look at the clonal level. And what we developed in the past years, that we developed barcoding systems where we can track individual clones and then see, for example, in such a spatial transcriptomic setting, we can read out the barcodes and then look how these barcodes are linked to specific programs. Here you can see this is a tumor that is infiltrated by neutrophils, but the other nodule is not infiltrated at all, which means is that intertumor heterogeneity plays an important role also in this immunosuppressive microenvironment. So these are MDSC high, is a MDSC high nodule, whereas this is not, which of course then also responds differently to therapy. With that, I want to finish thanking the group. So these people did all the work, and I would especially like to thank the collaborators, which are essential for all of our work. And you for your attention, of course. Thanks a lot.
All right, thank you. At this time, my pleasure to introduce Dr. Rory Smoot, who is a hepatobiliary and pancreas surgeon and associate professor of surgery, biochemistry, and molecular biology. He serves as the research chair for the Department of Surgery, as well as the director of the Surgical Clinical Research Office. Institutionally, he serves on the steering and executive committees for the Center for Biomedical Discovery and Center for Cell Signaling and Gastroenterology at Mayo Clinic. He has a research program that's focused on understanding the molecular mechanisms underlying the development and the progression of cholangiocarcinoma, as well as the molecular mechanisms that underlie liver regeneration, where he tests novel targeting strategies and therapeutics in preclinical models. His research program is funded by the NIH, the DoD, Regenerative Medicine Minnesota, and the Mayo Clinic. So, Dr. Smoot, thank you.
Thank you to the organizers for allowing me to talk about this unique ongoing project that we've had going for about 4 years, where we've been waiting for computing power and math to catch up. A couple things. This is the Plummer Building at Mayo Clinic. It's the original really the original clinical building that's lit up in honor of cholangiocarcinoma. They can change the colors, and we got it green, so we were very happy with that. So you've seen this figure several times today, and people know in this room that the classic classification for cholangiocarcinoma really is anatomic, intrahepatic, perihilar, and distal. But the only real therapeutic implication relates to surgery and transplant in that setting. Are you eligible for do you meet transplant criteria? What do I have to do as a surgeon? Is it possible to remove it? There are certain molecular subtypes that run with these anatomic subtypes, but not uniquely. There are molecular approaches that have been developed to try to improve classification, and certainly there are a few molecular drivers that are known. But most of these, even the multi-omic versions of them, tend to be these layered approaches where an omic is stacked on an omic is stacked on an omic, and we're looking at clustering and trying to look at activity. But for the vast majority of patients, they still do not identify targets. And so some of the issues that we've run into with these previous studies is really the inability to integrate the omics. And it goes a little bit to this right-sided thing here, the relationship between mRNA and protein. It's not a one-to-one relationship. Changes in the RNA do not dictate changes in the protein, and certainly not in the changes in the regulatory state of protein, activation, inhibition, which are incredibly important and are certainly the effectors of the effectors in the cell. So you've had, again, these kind of stacked clustering type of analysis, and you've had the sequence-centric analysis, but misses a lot of this effector function that we know the proteins really represent. So we asked the question several years ago, can we develop a functional multiomics classification utilizing newer bioinformatics techniques? The other thing I'm going to tell you as a disclosure, I don't like math. I went into biology and medicine because math was not my thing, so I will do my best to explain what we've done here, but we used a lot of math. And does this provide biologic insights and new therapeutic targets, things that we can do and go after some of these hidden signals that we've not picked up in previous classifications? So what did we do? So this is one of my patients. This is an operation that we did. This is a perihilar cholangiocarcinoma. We took 43 of these patients, relatively evenly matched in terms of gender. Importantly, and I think very importantly, because as Dieter had pointed out, in the field, there is very little information across the subtypes, right? And we went into this thinking that this is a molecular disease, not an anatomic disease. And so we included all anatomic subtypes. It's still, as most are, weighted a little bit towards intrahepatic. We have perihilar and distal. Our median survival was 31 months. We had a lymph node positivity rate of 29%. So we had 21% of patients that had multifocal lesions. So we had a good mix of anatomic subtypes as well as known clinical prognostic factors for these patients. What was our workflow? And really this was born out of a desire to really get at the proteome in these patients. And so we took the patient tissue, we did 8 of the patients, we took normal paired liver, did RNA sequencing, whole global proteomics. We did phosphoproteomics 2 different ways. We did a phosphotyrosine antibody enrichment. We also did IMAC, immobilized metal affinity chromatography to get at the proteome, and then some of our downstream analysis that I'll talk about here. So into our models, I'll say we had a little bit more features that came out in the global characterization, but into our models, we were able to put 24,000 RNA features, over 8,700 global proteome features, and over 26,000 phosphoproteome features. There was a large study in Cancer Cell, 2022, proteogenomic characterization, again, of only intrahepatic cholangiocarcinoma in an Eastern series out of China, out of Fudan. That was, and you can see we've got excellent coverage, but we also have a dramatic increase in the amount of phosphocytes, so the regulatory components that we picked up, including in that study they only identified 41 phosphotyrosine sites, and we picked up over 400 in our study, including 353 new unique sites. So we can do this. We can do the usual PCA plots, and you can see that the cholangiocarcinomas are generally, other than this one perihilar tumor, are clustering away from clustering away from the normals here, but that really doesn't get us much in terms of activity. And we can do these type of things where we look at volcano plots and say at a global level and a phospho level, what are we seeing up and down? And we see several targets that were picked up in that previous paper and multiple other papers. And while these are interesting from a standpoint of potential biomarkers and potential function, and we can do that, we really wanted to get deeper, okay? And so we wanted to understand the signaling complexity, and this is my favorite pathway in the world. Everybody at Mayo knows this, that YAP and TAZ and TEADS are important. Jennifer Tomlinson will present a little bit of our work tomorrow during an update for one of the fellowship talks in terms of targeting this pathway. But we always draw the pathways like this, right? And we always think about the pathways like this, but that's not what they look like, right? They look like this, right? So this is just the mTOR signaling network, but it is an absolute meshwork of interactions, and there's all kinds of regulatory aspects that can change. And so the question is, could we leverage big data? Could we leverage computing data or computing power to kind of try to help us understand these patterns, which we can't look at and understand? So one of the things we needed to wait for in terms of how to do this was the math. And so this is a relatively new technique. It goes by MOFA, Multiomics Data Integration, so Multiomics Factor Analysis. This is a joint dimensional reduction approach, okay? So think about a PCA of a PCA of a PCA. So this is PCA on steroids. It's going to just help us find the heterogeneity in the data. That's all it's trying to do for us. It finds features associated with variability in the data. The variability I'll show you are called factors, and within those factors we see our molecular features, protein components, phosphoprotein components. You can incorporate multiple normalized omics outputs. It does work best with data with Gaussian distribution. There are ways to try to put genomic data in there, but it's a little bit more finicky, and so we stayed away from the DNA. We used RNA, protein, and phosphoprotein. And it's very important. It finds variability in the data. That's all this is meant to do. So you need to ensure there's no technical or batch effects in the data set before you go forward. So what does this look like? You get the MOFAgram that comes out here, the factogram. So we get a factor value for these. And so the algorithm runs, and it finds degrees of heterogeneity. And it will run and find factors that will contribute at least 2%. So it'll run out until you've basically maximized any heterogeneity. So it'll find heterogeneity underneath their omics. It's incorporating all three. Importantly, in our analysis and the power, to try to demonstrate the power of the multi-omics approach, each one of these dimensions of heterogeneity was made up of all three omics. There was not a single omic that took over any one of the degrees of heterogeneity in this. So all components were necessary in order to see this. We then took this and applied a phenographic approach to these factor values to obtain clusters. And so what does that look like? So here are the clusters, clusters 1, 2, and 3. We didn't get very inventive with this, but a couple of really important things that come out of this. You can see that while cluster 1 and 3 are represented by all of the factors, now again, the heterogeneity is defined by all factors, but when you look at the actual clustering, you see that cluster 2 itself is only defined by protein features, even when the model has the ability to look at all the RNA features. This cluster will only be picked up if you're looking at all three omics at once. Why is that important? Because they are different. Cluster 3 pathways map to chromosomal stability, apoptosis, cell cycle, regulatory pathways. Cluster 1 are almost all immunomodulatory pathways, and cluster 2, the protein-based, are all metabolic pathways, which is quite interesting from that standpoint. Here's the other real big takeaway from this, is that The anatomy does not matter. This is a molecular disease. You can look across all clusters and you can see that the perihilars, the interhepatics, and the distals are spread. Now we only have 2 distal tumors, but are spread across these clusters. So there's a unique set of molecules that each one of these tumors has access to co-opt, to grow, and it does not matter, the anatomy does not matter. So what do we do with this? We can see some biologic significance here. All of these patients were resected for cure with curative intent. There was one that went in with curative intent, one had a palliative procedure, 2 patients had perioperative mortality. When those were removed, you can still see that cluster 2, that metabolic cluster, is significantly worse than either cluster 1 or 3. So there is some biologic significance to this. This is an unsupervised approach that got us here. What do we do with this? Well, we want to find targets. What's happening? How are these different? So we look at kinase enrichment, and we can do this a few different ways, and you can see how this is representative. We're looking for basically, we can rank the kinase activity based on what we're seeing at a phosphoproteomic level onto a single dimension, and then use Euclidean distances to try to tell us where, what cluster do these map to and what's important. There's 5 kinases that are highly ranked, meaning appears to be very active in all of the clusters. AKT, we just heard about that, CDK1 and 2, EGFR, which there's a lot of interest in the field based on some recent research, published research, and GSK3 beta. So one question is whether or not those kinases represent early changes, as they're in all of the clusters, versus some of these more cluster-specific kinases, and then just non-ranked cluster or kinases. And you can see an example here of some of the kinases that we see across there. Well, we find targets, we got to validate. So one of the things we do is we take our PDX models and we map them back. So we have 28 of our biliary tract cancer models that we have taken through this multiomics approach with RNA, protein, phosphoprotein. We developed a bioinformatics workflow with a vector machine approach, a machine learning approach, to map these back on to the clusters, so we can say which one of our PDXs are cluster 1, 2, or 3. We go back to our kinases, and you get an example of the ranking system. It's golf, low is better. And so you can see an example here. We'll look at cluster 2, the metabolic cluster. A kinase C, for example, has a very low number, highly ranked, as opposed to 500's the top, so cluster 1 doesn't seem to care at all, and cluster 3 is kind of intermediate here. We pulled a cluster 2 PDX, which is an IDH mutant, which is nice to see that that's the one that, that's one of them that mapped to cluster 2, that metabolic. And you can see here, using a small molecule inhibitor to that kinase, we can see efficacy in that PDX model. We have several additional targets and several additional models that we're looking at, including looking at the differential between these types of, like a cluster 1 PDX with this treatment. The next step that we took was this hierarchical all-against-all association testing, and this is to try to get back at some of the pathways and molecular features that are important clinically. And this is, again, part of this waiting for the math to catch up. This is a technique for evaluating high-dimensional associations, okay? So it allows us to develop biologic inferences from the MOFA output. So the MOFA factors can be compared to a metadata variable, lymph node metastasis, overall survival, that type of thing. Response to therapy in the PDX models, you can map back with this, which is fantastic because you can find some biomarkers of potential response. I'll show you that. But the factors associated with the clinical data point can be different for a different cluster. So in other words, there can be multiple molecular routes to an outcome. Okay, so depending on what cluster you're in, the types of features associated with, for example, lymph node metastases will be different. I'll show you that. Oh, the bioinformatics team, this is striking to me, and so I always put it on here. The first time we ran this algorithm is 30 trillion comparisons for this data set for 43 tumors across these omics. It took a week to run on a cloud computer that is 50 million comparisons a second for an entire week. People don't understand how big 30 trillion is. So 30 trillion comparisons to try to get us to some output here. we crashed the server the first time we did it. We got in a little bit of trouble, but it's running now. And this is an example of the structure that we get out of this. So this is molecular features associated with lymph node metastasis. You can see here the blue patients had pathologically confirmed lymph node metastasis, and you can see some of the structure here in terms of the molecular features at an RNA, protein, and phosphoprotein level. And when we collapse that into pathways, I don't know how good that's coming across, but you can see the structure and the data here with two very unique sets of pathways associated with lymph node metastasis. And now we're going back and looking at a few of these negative ones to see the survival outcomes for these patients, because we would predict that they had very poor survival, and they may have had multifocal disease as well, based on what we're seeing. So the last data slide, just to show that, again, what I was talking about, now that we have this data generated and these models generated, one of the things that we do in the lab is when we get new small molecule inhibitors, for example, new therapeutic approaches, we go back to the PDX. We, of course, can't look at immune-targeted therapies, but we go back to the PDX, and we treat generally somewhere between 4 and 6 of these models. We've done this with GemCis. We've done this with several small molecule inhibitors. And then we can simply map back to the molecular features associated with sensitivity or resistance. I can tell you the types of features. We say drivers, but these are really associated. We don't know if they're actually driving the resistance, but I can tell you the pattern for patients, or for tumors, PDX tumors that will respond to gemcitabine cisplatin, will respond to, this is a SARC inhibitor, which is nice to see that several of the SARC kinases are showing up on the sensitivity signature. And then we've got another one that's a GCN2 kinase activator that we've run through as well. So in summary, a quick rundown through a long bit of work that we've been doing. But the integrated multiomics provides a functional classification system identifying novel therapeutic targets. These data-driven approaches provide insight into tumor biology, help us deconvolute this very complex pattern for these tumors, and they can be leveraged in combination with preclinical model treatment studies to identify biomarkers of response. So I need to thank the team. We have a multidisciplinary liver research program at Mayo Clinic. It has, I can't remember, it's 15 or 20 PIs, but within that there's a subset of us. There's 7 of us that work on cholangio. We're missing Lewis. I don't know where he was that day, Lewis Roberts, but we have a multidisciplinary team. Myself and Suzanne Warner are hepatobiliary surgeons. Caitlin Comboy, who's here, is a medical oncologist. You heard about Samara, transplant hepatology, transplant oncology. Greg Gore's transplant hepatology, and Davide Povero is a cancer biologist in our bigger team with our funding there. And I was told I needed to put my contact info up, so we'll go back to the Plummer Building picture. Thank you.
Dr. Schneider, yeah, if we could have all the session speakers up at the front for the panel discussion, and we'll open it up for questions. So if you have questions, I'll come around to you. Dr. Schneider, you have a question.
Yeah, I do, for Rory. Is it on? Okay, is that on? So Rory, that was a great talk. So did the data point you to any new targets that were unique to this cancer?
Yeah, so
Past kinases.
yeah, so we focus on kinases just because they're targetable, and we can see it in the post-translational modifications, right? And we didn't look at assimilation, we didn't look at palmitylation. Right? We heard last week at ACR there's a single protein that has a purification or something like that. That's a new one. So we focus on the kinases. What I would say is, like, for example, the one I showed, that kinase, that is unique. It's only been looked at in a single study in histological malignancies. So we found, for each one of the clusters, we found half a dozen or more. One of the issues that we've got into, which we need to talk to you about afterwards, is that we've been going after some of these targets, like for example, YAP and TAZ, which don't have direct inhibitors, with nanovesicle-mediated like siRNA approaches to try to target some of these things that can't be dealt with at a systemic level. So that's the one risk we run with these kinases, is I think the off-target effects and the toxicity potentials are relatively high there, but we have a list of a dozen or so that have not been looked at in this.
Are you thinking about developing high-throughput screens to maybe look at new chemical matter?
Yeah. So
because currently you're just looking at FDA-approved small molecules?
So we've mapped back to we've mapped back to FDA-approved molecules. We've looked at tool compounds that have been published for some of the kinases, but we absolutely need to, now that we have some targets, that would be the next step, is to look at to do some high-throughput screens for sure.
Thanks.
Ben, I'll come around to you.
So is it a question for Greg? Can you give us a little bit better, so the CD26, CD4 cells are where you're banking your money for now. So can you give us a better sense of how you go from a pheresis product to an infusion product that's enriched for those particular cells?
Yeah, we've done some of that work already, not with pheresis products, but from peripheral blood, from TILs. You can indeed expand them out. First trick is to sort of, select them. And then there's an expansion protocol, usually involving beads that contain, the key is really giving ICOS stimulation to them to really make them go. Then you can, even if there's a few, you can make tons of them out of it. So that process in preparation for the trial, we're doing all the pre-IND workup now. And obviously the key component is showing that you can expand and then isolate them and demonstrate some functional activity once they're antigen redirected with a CAR. So there's sort of a cocktail that we do, and incorporating the duvelisib, the PI3 kinase inhibitor, slows down the yield a little bit, but it also makes them of a higher quality. And there are estimates that this is indeed feasible from a pheresis product in terms of getting enough cells, which is obviously a big barrier when you're talking about selectively identifying a cell population to put back. So thank you for that question.
So I have a question for Dr. Sauer. So that was beautiful work, and the cancer discovery paper, it clearly shows that tissue specificity matters, context matters, and to do it, for perihilar cholangiocarcinoma and distal, I think that's a significant contribution. There's a recent paper, you may have seen it in JHAP, P10 deletion in the extrahepatic biliary epithelium leads to bilin-like lesions, and with aging, invasive cancer, and that's exacerbated with P53 loss. And you had mentioned P10, but I wasn't sure if you had looked at P10 deletion and whether you observed a similar paradigm with P53 deletion that exacerbates the bilin lesions.
So we also deleted P10 alone and in combination together with Owen Samsung. He did in Glasgow these experiments, and we see an acceleration. So we see tumor formation alone. So if we delete P10, as well as in combination with KRAS, as well as in combination with PRCK activation, and P53, I think it's a global gatekeeper across all of our models. So we, if we delete it, we can we see acceleration in all of the different organs. I think there's not really tissue specificity anymore. Then, thanks.
And what do you think? Do you have a hypothesis, or what do you think is the mechanism behind the tissue specific, the differences, right? Why did context matter when it came to KRAS? because you didn't observe it, in the biliary epithelium, but it clearly leads to cancer in pancreas. Do you have a sense of what is underlying that difference?
I think the tumor suppressor pathways are very different in different tissue types, and that depends, of course, also on, for example, on the stem cell properties of the cells that are targeted. For example, in the intestine, you have highly proliferative cells, therefore they do not have a kind of a senescence program, whereas in the bile duct or in the pancreas, proliferation is very tightly regulated, and therefore also the tumor suppressor programs are very distinct, and the tumor suppressors that are active in different tissue types are very distinct. And we have to break them down, so to say. And PSAK is able to downregulate P27 on its own, whereas in the KRAS model we need a kind of a secondary genetic hit that then targets somehow P27, activates PSAK, for example, signaling or other pathways.
How important do you think oncogene-induced senescence is in cholangiocarcinoma, or is it?
I think it's an important tumor suppressor pathway, as it is in the pancreas, but there's still specificity for that.
Other questions?
This is for Dr. Schneider. Can you put your hydrogels on stents? Have you tried to put them onto a stent, or either like a covered metal stent or a plastic stent, as a way to perhaps treat biliary tract malignant obstructions?
Yeah, we have not used our gels for coating stents. There's a lot of good technology that's available to coat stents, and I can point you to that literature and those technologies. It's basically functionalizing the surface of the stent with whatever biomolecule you want, and there's even technology where you can release those molecules as a function of time from the stents currently. So there's really good technology available.
That'd be very interesting, yeah. And then for Dr. Sauer, have you isolated cells and tried to re-implant them into the biliary tract?
Sorry, I didn't.
Have you isolated cells from your animal model and then tried to re-implant them into the biliary tract?
Yeah, we have done that. I mean, it's difficult to implant them into the extrahepatic bile duct, of course, because there's not really tissue available. But we did it then by transplanting them into the gallbladder, for example, and that's possible, and they engraft there as well. So we isolated organoids from the tumors, and then we implanted them. And I think that's a nice possibility now to have more robust models and quicker models to test therapies in an immunocompetent setting, because we can transplant them into syngeneic mice.
So you put them into the gallbladder. Did they get jaundiced?
Sorry?
Did the mice get jaundiced?
Yeah, they get jaundiced at late stage disease.
Other questions?
Thank you. That was a really interesting session. I just wonder whether the panel generally could comment on whether we've missed AKT and PI3 kinase from only looking at genomic mutations in the past. PI3 kinase mutations are very rare, but it seems to me that across intra and extrahepatic, this pathway is a major driver. And also, how does that then feed into some of these changes that we see, kind of both common changes in intrahepatic versus perihilar disease and the differences?
Yeah, I think that this is a little bit of the concept of the hidden signal, right? Is that when we're only looking at genetic changes, mutational landscape, we miss, and even just levels, we miss activation states that can indicate that it's highly important. I mean, our data set would suggest that 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 activation pathway is, that pathway is active in a majority of the tumors that we looked at. And I will tell you, so 43 patient tumors, 28 additional PDX, we can see it there. We clearly generate tumors with AKT. It's permissive for sure, and so I think that is an important target in this tumor, and I think that it's across all subsets of anatomic subtypes.
Yeah, I agree. We have to look beyond pure genomics and look at pathway activation and also co-activation of different pathways. We, for example, see that in a KRAS setting, if we overactivate PRCK, then we get whole genome duplication and genomic instability. And such processes are not really linked then to a mutation. It can be then also copy number variation, PTEN loss, and so on.
Yeah, so one of the things that we've done that I didn't show here, but we'll include is we use the HALA approach, essentially to say, when you have a genomic alteration like a p53 mutation, KRAS mutation, what are the downstream multiomic features associated with that? I mean, because again, at some level, in order to operationalize this, we need to go back as high as possible up the chain so that we can find what the diagnostic is going to be. But there's going to be unique changes, and when we map back to clusters, for example, p53 all goes to cluster 1, 67% of KRAS mutants go to cluster 2, I think. The HER2 amplifications go to cluster 3, but it's not 100%, and so even with these very strong drivers, there are these other downstream modifying features that change the output.
So fascinating talks. Thank you all so much. I have two questions. First one for Rory, and the second one combined, Rory and Greg. So fascinating data. Just curious, in terms of the technology that you're using and the cost and all that, what is your thoughts on the eventual applicability and reproducibility outside of Mayo to validate these efforts in a larger scale, and again, in a generalized population? And then the second question, in my own naive understanding of this, we have amazing immunotherapy efforts, both in the preclinical in the lab and in trials, and then we have targeted therapy efforts, both in the lab and in trials. But I feel like they both run in parallel right now, both again in the lab and in our clinical trial space. What is the horizon, or what is your vision of eventually combining these two efforts, both preclinical and in the trial space? Because maybe that's the ultimate personalized therapy is immunotherapy with targets. So, anyway, fascinating talks. Thank you.
Yeah, so this was not cheap. You can imagine to do this. This was, I think, $700,000 or $800,000 to do, and that doesn't count the time trying to sort it all out and figure it out just to do it. But a couple of comments there. One, we are now rolling back bioinformatically and trying to determine the minimal classifier set for each thing that's proteomically based, okay, because we can go out to data sets and at least try to see how often do we see the data, those subtypes across the data sets. And what we're really fascinated to do is actually look across other tumors as well, because again, this is an epithelial adenocarcinoma, okay? And sure, there are tissue-based contexts which may be epigenetic, but epigenetic changes are then, when you feed forward, the differences are in the proteins, right? The proteins are your effectors. Now there's metabolomics, there's all kinds of other contextual issues that have to be considered, but I think we'll be able to apply these models actually across other, at least epithelial-based adenocarcinoma, so we want to do that. The second comment I would have is that from a clinical standpoint, like when we operate on somebody, right, and maybe economics isn't the best thing to talk about, but it's $100,000 for one of these, big operations and the hospitalization. A round of proton beam therapy is going to be closer to $200,000. There's been a couple of economics papers. When you get to third-line systemic treatment, it's $33,000 a month, and that doesn't count the hospitalization for patients. So the concepts that we're not willing to use all the information, when a patient comes to me, we use everything possible to make the decisions. We're doing incredibly delicate and high-risk operations and other therapies, and yet we step back when it comes down to Would I want to know every possible thing about that patient's tumor? And quite frankly, not just one snapshot. I want to know what happens under therapeutic stress as well. How does the landscape shift? Because that landscape's going to shift as soon as you put them on a targeted therapeutic. There's going to be new synthetic lethalities and things like that. So I think there's going to need to be something that happens from a standpoint, and I realize that you can't just throw everything at everybody, but we have to be willing to dive very, very, very deep on some patients. And I think that this is a great disease to do it in, because there's not a lot of good options, in terms of treatments, and it's a rare disease. So I think we got a lot of room to make, and again, combining that with rational approaches for clinical trials, I think is going to absolutely be necessary. But also probably suggests, at least from our data, is that you can't combine all of these together. You lose the signals, right? If you combine, and I know why we do it, because there's not enough patients. But when you combine everybody with a biliary tract cancer into one lump and then do it, you lose the signals. So.
And I'll pick up on your second question, related to personalizing immunotherapy. I think that's exactly a great idea. And one of the things you could argue within cholangio that has been the most impactful has been really genomic characterization of disease. Look what's happened with FGFR fusions and changing the paradigm of clinical care for those patients, IDH mutations. And certainly the next frontier could be merging our knowledge of what the genomic feature of a cholangiocarcinoma is with what that tumor immune microenvironment looks like. And we're already seeing blips of this with IDH mutant due to work out of Nabil Bardisi's lab and others that define a particular immune profile of those tumors. And as we continue to slice out the pie of what the genomics of these individual cholangios look like, and really all biliary tract cancer, put gallbladder in there, I think we'll have a lot to learn about which cells dominate which tumors and what potentially targetable immune markers they express. And I think the field's just in its infancy. And I really enjoyed the idea that Dr. Smoot proposes it's not necessarily driven by anatomic subtype anymore, it's all molecular. And I think those are some really important information and speaks to the genomic heterogeneity of this disease across the board, and that could certainly apply to your comment with regard to the immune features as well.
All right, I think with that, we'll conclude the session. Thank you, everyone. That was a really great discussion. We have a very brief break now, so we'll reconvene at 10:10. So we'll see you then.